So the work I will be talking about today is very much still in its early stage, but I'm very excited to share with you some preliminary results. So skin cancer is the most common cancer type in the United States. In fact, the number of skin cancers diagnosis outweighs that of all other cancer types combined. So among the three major types of skin cancer, melanoma, which starts in the melanocytes in the epidermis, only makes up about 2% of the total cases of skin cancer. However, it is the most aggressive, with notably higher rates of metastasis causing up to 60% of skin cancer mortalities. For metastatic melanoma, besides surgery, there are varieties of FDA-approved targeted and immunotherapy strategies available to patients. However, the degree of response to therapy varies from tumor to tumor. This difference in response can be in part attributed to the molecular heterogeneity within the tumor. For example, treatment resistance and relapse may be caused by the presence of a stubborn subclone. So if we can better characterize such intratumor heterogeneity, we may improve patient outcome. A prior study by Ender and colleagues in 2016 characterized the degree of intratumor heterogeneity across 12 cancer types. They found that among the samples in consideration, on average, melanoma samples have the highest number of detectable clones, indicating a high level of intratumor heterogeneity, which we set out to study in our work. How such heterogeneity arises can be modeled as an evolutionary process. And to study such process, a common analytical approach is phylogeny reconstruction, which, given a sequenced tumor biopsy, reconstructs the tumor phylogeny based on the observed somatic alteration events. And from the reconstructed tree, we can infer the evolutionary trajectory of the tumor. So in this work, we want to better understand melanoma intratumor heterogeneity via phylogeny reconstruction, specifically leveraging the advantages afforded by data generated with Nanopore long reads. So for data, we make use of a genetically engineered mouse model published in 2020. In particular, we used the M4 mouse melanoma model. Evaluation of the mutations and expression profiles of the M4 model suggests that it corresponds to the RAS mutant transitory human melanoma in the Cancer Genome Atlas. So the M4 model is interesting to us because it seems to harbor heterogeneous cell populations that resulted in differential response to treatment. So here, a cell line was derived from the M4 model. Allografts were derived by injecting the melanoma cell line in syngeneic mice, and these mice were subjected to immunotherapy. As we can see, while 60% of the cell line-derived allografts showed no difference in tumor growth compared to control, 40% showed tumor growth delay. This is interesting to us because it recapitulates diversity in response to CTLA-4 blockade in human patients. We would like to characterize heterogeneity of this melanoma model by reconstructing its clonal evolution history. So to generate the data we used for analysis, 24 single cells were extracted from the model-derived cell line culture. Each single cell is then cultured into its own subline containing a cell population that's relatively genetically homogeneous. Each subline sample is then subjected to Nanopore long-read sequencing. Even though the data for each sequence subline is not of single-cell resolution, because each subline population is relatively homogeneous, important genomic features should be stable within this subline population. Our goal is to use the Nanopore long-read sequencing data to infer the phylogenetic relationship among the cells from which the sublines are derived. So in our work published in Genome Research earlier this year, we showed that it is possible to leverage genomic methylation profiled from single cells to construct tumor phylogenies. And to obtain an accurate tumor phylogeny, the key is to distinguish CpG sites with phylogenetic information from those whose change in methylation status are more akin to stochastic noise. So now the question is, can we adapt the CpG methylation-based phylogeny reconstruction algorithm for methylation profiled from Nanopore long-read sequence sublines? So it turns out the ability to perform haplotype phasing with Nanopore long reads and subsequently the ability to obtain haplotype-specific CpG methylation information is a great asset in distinguishing signals from noise when we try to build methylation-based phylogenies. we put together a computational pipeline to obtain haplotype-specific CpG methylation information from the sublines, as well as a normal control. So here's a diagram visualizing what happens. For each sample, once the reads are assigned to a haplotype, since we can simultaneously profile CpG methylation from Nanopore long reads, suppose we have two CpG sites of interest, denoted by the two red vertical bars, we can derive allele-specific methylation frequencies from the reads assigned to either haplotype. We examined how the methylation frequencies for each CpG site compare between two haplotypes. So here on the right is the result for a particular subline. The x-axis contains bins for methylation frequencies for one haplotype, and the y-axis for the other. And the heat map counts the number of CpG sites with respective methylation frequencies along either haplotype. The high counts on the bottom left and top right indicate that a majority of CpG sites have consistent methylation status within reads tagged with a particular haplotype, and that the methylation status are usually the same between the two haplotypes. The colors at the top left and bottom right in the heat map are also worth noting. They show that there are a decent number of CpG sites with consistent methylation status within either haplotype, but the methylation status is different between haplotypes. For each subline, we retain the CpG sites that fall at the four corners of the heat map from phylogeny reconstruction, as the rest have inconsistent methylation status even within a haplotype, and therefore are more likely due to stochastic noise or error. The sequencing of the sublines is still ongoing, but we have some preliminary results on 8 available sublines. We did some additional filtering based on read support and computed pairwise distance among the sublines, which we define as the number of CpG methylation status differences between the haplotype-specific methylation vectors of the sublines in the pair, and then we normalize it by the number of non-noisy CpG sites that they share. Here is the unrooted tree we obtained by applying neighbor joining to the pairwise distance matrix. We will be able to root the tree once we obtain data from the normal melanocytes. To validate this phylogeny, we looked to our previous work that profiled the sublines using bulk whole exome and single-cell RNA sequencing. Specifically, we looked at the phylogeny constructed by the mutations called from the short read sequencing data. So I've boxed out the corresponding sublines here on the left, and you can see with somewhat limited resolution, the phylogenetic relations inferred by the two methods here are largely similar. Here we have C17 and C14 as outgroups, C1, C4, and C9 being nucleate, and C11 and C13 are nucleate. That said, the phylogenies do not completely agree, and it is evidently noted by the placement in C8, which share clade with C11 and C13 on the left, but is further up in the phylogeny on the right. Of course, these are very preliminary results, and we will be able to look deeper into the disturbances once we have all the sublines, as well as normal melanocyte data. That said, the initial results seem promising, and what made us more excited about this project involving Nanopore long read is that we can perform small and structural variant calling on the same set of data. This effort is led by our collaborators. Anton is performing small variant calling with deep variant, and Aisha is leading the development of a structural variant calling tool called Cerberus, which we'll hear more about in Misha's talk. Here you see the upside plots showing respectively the number of small and structural variants shared by the subset of sublines. The ability to simultaneously profile small variant, structural variant, and CpG methylation from the same set of long read data presents an unprecedented opportunity to harvest additional signals for integrative phylogenetic analysis. For example, with the currently available data, Cerberus is able to identify a contagious event followed by a loss of heterozygosity, as you can see here. So in C1 and C5, there are a set of localized hypermutations and a high degree of heterozygosity. In C13, however, while we see a lot of the same mutations as in C1 and C5, one allele seems to be lost. This set of observations would suggest that a phylogenetic relationship among the three sublines is shown on the bottom right, with the two genomic events placed as such. So, of course, this is only one example among many others we're still in the process of analyzing. We plan to construct phylogenies using only small variants and then only using structural variants, compare them with a methylation-based phylogeny, see if there are inconsistencies or ambiguities, then see if an integrated approach leveraging phylogenetic information from all three sources of signal could resolve some of the ambiguities in an improved phylogeny. So this brings us to the end of my talk. In summary, we generated a Nanopore long-read sequencing data for 24 single-cell derived subline from a mouse melanoma model, and we hope to leverage small variants, structural variants, and methylation jointly profiled from Nanopore data in an integrated phylogenetic analysis, which will hopefully give us a more holistic view of melanoma evolution and treatment resistance. So with that, I'd like to acknowledge my collaborators at both the National Cancer Institute and the University of Maryland. Thank you so much.
Thank you, Lynn, for this great